So this was a great morning this morning and very stimulating. And what I'm going to try to do is, I realize this is a mixed audience, so I want to give you a little bit of appreciation for what's going on with DNA and genetics these days and how we're trying to integrate this kind of information both for research purposes and for clinical purposes into our very large health system. I do work in the Feinstein Institute, which is a separate institute within the health system, has its own board, and has an incredibly collegial atmosphere of very smart researchers there who are willing, like Michael, to take a chance and leave the big medical centers and come to this place, which is really very swift on its feet and willing to take chances on new projects, and I'll mention some of those. So, DNA sequence, as you know, is information. And the situation now, as opposed to, a decade or two ago, is that getting the information in terms of the sequence out is a trivial matter. And it's increasingly less expensive. So you can now do a whole genome for $1,000, $1,500. But understanding the information is another matter entirely. There are lots of very smart informatics people working on trying to parse these data and link it to disease and phenotype. But there's still a ways to go. And what I want to mention is that we need to understand how the cell uses the information in order to really understand what the information is, because the pure sequence alone, no matter how fancy your algorithms, requires you to understand how the DNA is organized by the cell and how it's annotated by the cell. So let me just refresh your memory about DNA. DNA is actually physically 1 meter in length, and it's packed into a nucleus of a cell. Every cell has its nucleus, so that's 1 to 1,000,000 packing. And to just give you a scale of that, 1 to 1,000,000 packing, if you scaled up the DNA molecule, which is divided into 23 chromosomes but is 1 meter long, to a size 46 thread, which is about 0.2 millimeters, it would go across Africa, and that would get packed into a 4-meter diameter ball. And the cell is asked to extract 3.2 billion bits of information out of that ball. So you're faced with that ball, and you have to get that information out. That's obviously a huge task, and there's a lot of organization that underlies what goes on with packing this, the packing of the DNA. So the first level of packing is important to know about is around nucleosomes. It's kind of a beads on a string approach. This was discovered in the 1970s, that this is the first layer of organization, and then there are subsequent layers of packing until you get to the entire chromosome, which is a lot thicker and more densely packed. And this packing involves circling about 150 base pairs of DNA around 4 different histone molecules that each can get coded in different ways to tell the cell, should I be using this piece of DNA now, or should I not? So again, the organization at this first level is telling the cell, is this DNA information open for business to the cell or not? If it is open for business, it also gets bound by other proteins that either copy it or block it or modify it in some way that is additional information about what the cell is doing or what the cell should be doing. So actually, even though the human genome was sequenced, in 2002, and we all think of it, well, we now know what the sequence is, we've learned a lot about its variation. What we still don't know about is how the packaging occurs and the details of this packaging and how the information is utilized. That's been the topic of the ENCODE project, Encyclopedia of DNA Elements, which is an international project, to define for every different kinds of cell, how is this packing organized? So this is really the field of epigenetics, figuring out what part of this molecule of the genome is actually open for reading and for information transfer to the cell. And that, most of the ENCODE approach to this has been very labor intensive. uses very large amounts of DNA, and is very slow. That is about to change because there is a new technique called ATAC-seq in which you can actually take a very small amount of DNA, very small numbers of cells, and expose it to a transposase, and that will put little markers around the open chromatin, and then you can screen very quickly across the entire 3.2 billion base pairs of the genome what's open for business in that cell. So this is a paper that was just published on T cells. There are 66,000 areas that are open for business. That's only a few % of the genome. Most of the genome is closed for reading. And what's interesting is that a large fraction of that, some of these elements, about 4,000 in this case, are variable either between individuals or within an individual over time, or between genders. Very interestingly, there are big gender differences in how the genome is open for business in females versus males in CD4 T cells. So this opens up a new world of thinking about interrogating the genome for not its sequence, but for how it's differing between individuals, how it's differing within an individual over time, how it differs on smoking, on exposure to pollution. All kinds of environmental exposures are going to influence this, and it means that we have a potential measure not just of your inheritance, but of what your genome is doing biologically now in a given cell. So I think this is, indicative of a new wave of thinking about genomics, which actually adds a whole other level of complexity than simply associating the sequence with a particular phenotype or disease. So the rationale and approach for integrating either genetic or epigenetic information into a health system, one approach is to link it with information that's in electronic health records, and also have tissue biorepositories and longitudinal data about outcomes, and try to integrate it into that. So using that approach, you could discover new disease association, predictive markers, and obviously cancer and now pharmacogenomics is a good example of the success of that, although still very, very early stages. And epigenetics, is just starting to be integrated into cancer. It's basically observational. And you need to figure out how to integrate all this data, get it in one place so smart people who know how to analyze these complex data sets can pull out meaningful associations. So that's one thing that we are starting to do, and many other health systems around the country, including Kaiser Permanente, has done, and they're mining that data. The thing I want to emphasize that we're doing that's a bit novel, I think, is to not do purely observational analyses, but to actually create a living biobank. This has been done at the University of Cambridge and a few other places around the country in which you enroll subjects, they may or may not be sick, and be able to call them back for biological analyses, functional studies, based on their genetics. And I'll give you some examples of that. So I've been working on rheumatoid arthritis genetics for several decades now. We now have 100 genes for rheumatoid arthritis. And each of these peaks here reflect the statistical significance of that association. Some of them are extremely strong, such as HLA, which is, p-value of 1 over 10 to the 250th. There are 200... 10 to the 80th molecules in the entire universe. So this is incontrovertible evidence of association. So what do you do with this information? And I could show this same kind of slide for almost any other autoimmune disease, for Alzheimer's disease, for schizophrenia. They're all coming up with this model of many, many genes with extremely modest effects. Effect sizes often much less than 1.5. And that means that despite our thoughts when we started this, we're not getting diagnostically or predictively useful markers out of the genetics per se. You can't diagnose RA with this. you can't even really enhance the probability of your diagnosis knowing this kind of information. And the other part of this is that all these variants are pretty common in the population. Everybody in this room has multiple ones of these variants. So they're here for a reason. They've been selected for. They've also done good things in the setting of the population. So is this completely hopeless? Well, there are some examples that have come out of GWAS that I think are illustrative of how one could potentially use this information. This is not the typical result, but so, for example, we have an association with something called TYK2, a tyrosine kinase molecule that's involved in cytokine signaling for various important cytokines for disease, IL-12, IL-23. And this variant that's associated, the rare variant, which is about 3% of controls, is actually protective for rheumatoid arthritis, lupus, inflammatory bowel disease. So what's the function of this? Well, it turns out that these protective variants actually don't work well. They don't signal well. They're actually amino acid changes in this molecule so that when IL-12 hits the receptor, there's just not an efficient transfer of signal to the cell. So you might think, gee, this is a good drug target because if we inhibit this, it would reduce the risk of those diseases and maybe even treat them. But on the other hand, if you completely eliminate TYK2, either from a mouse or a human, you have severe immunodeficiency. So there's clearly a balance. So the question is, well, is this really a benign variant or is it not so benign, it just happens to be protective for RA? So this is part of a large family of signaling molecules that involves something called JAKs, Janus kinases, and TYK2 for some of these molecules. And in fact, there have been some molecules developed that are very successfully inhibiting these signaling molecules. There's no TYK2 specific inhibitor, but tofacitinib is a new molecule for RA that really does work. It comes with side effects. There's infectious disease side effects. There are some hepatic side effects of taking this. But in general, it's FDA approved, and it is used under the right circumstances. So the question is TYK2 a better target? Could you inhibit TYK2 to the right degree and not have these side effects? So in order to address that, we and Robert Plenge led this study where he looked at 29,000 subjects over 500 diseases in an EMR in Boston and asked, TYK2, which is present in 3% of the population, is it associated with any of these other diseases? Is there anything bad about having TYK2? And the answer was no. The only thing they saw was that they confirmed it was protective for RA. So I think this is the kind of data that one can extract from integrating genetics with an EMR and information about disease to get some insight into whether these normal polymorphisms that are present in the population are actually reasonable drug targets, even though on their own they have very little impact on the overall population prevalence of the disease. So of these 100 hits, actually very few of them are like TYK2. They're not amino acid changing. They're outside the coding region of genes. And they regulate, they are involved in the regulation of those genes. How much is made in what cell, in what circumstance. And as a result, it's very difficult to extrapolate from something that's just a quantitative thing to the entire disease. So it really has changed my thinking about how one looks at the function of such alleles. The traditional approach is to take the phenotype, which is usually very complicated. I mean, rheumatoid arthritis, lupus, all these diseases are complicated. And what we've done generally is try to define it as finely as possible, genotype, tens of thousands of people and ask about associations. There's always an environmental component. You try to control for that. Of course, this environmental interaction with genes has occurred over time. That's why these genes are in the population, because they've actually conferred some advantage. And there's always this problem of chance and fate. That's why twinning studies for most of these diseases far less than 50% concordance in identical twins. So the problem with this is that these associations that I've shown you are weak, and the phenotype is very complex. The alternative approach is to try to identify quantitative traits, what I like to think of as immune rheostats, that are actually regulated by these genes. And these quantitative traits are what predispose to disease. And these quantitative traits are going to be a lot closer functionally to the genetic variation than they will be to the disease as a whole, and will be a lot simpler. So this is an approach, so this is based on the whole idea of, stimulus response. In the immune system, it's very well shown that if you regulate the amount of something on the cell surface or in the cell, it really changes the stimulus response pattern of that cell. It goes through the whole immune system. And what most of these associations are doing is changing where this curve is. And so the question becomes, what does changing that curve do to a particular immune trait? And rather than asking about what it does to the disease as a whole. And this is just, I could show innumerable ones of these. This is just signaling pathways for T-cell receptor. There are about 5 or 6 genes in here that are associated with autoimmunity and immune function. Most of them are regulatory. They're not a structural change. So the question is, how can you ask what those regulatory variants do to T-cell signaling or B-cell signaling? And so, again, just as an overview of how we get at the problem of what these 100 genes are actually doing, if we know what they're doing, then we can start thinking about developing drugs that either enhance or inhibit what they're doing. But without knowing this, you really can't intelligently design interventions. So we obviously need to identify all the candidate causes variants. We're close to that. We need to understand what cell types they interact on, they act in. There's actually we have a lot of data now suggesting that you can take mouse models and actually, even if you can't transpose it directly to the disease, you can certainly transpose it to these immune quantitative traits or immune rheostats. So for that reason, about 10 years ago, we started at the Feinstein Institute, the Genotype and Phenotype Registry, which is very similar to the Cambridge Biobank bioresource. We have 5,000 people in this. We've got NIH funding to expand it to that level. We do 200,000 markers, actually now 1 million markers on everybody that enrolls in this. And we have the ability to call back people for a blood draw without IRB approval for studies. And we do this as a service to investigators around the United States. So if somebody comes and says, I have these variants I think affects T-cell function, but I've been unable to prove it. I put it into a cell, it looks reasonable, but I need to know that it actually does this in a living human being who does not have disease and who is not taking any drugs, because otherwise I'll be confounded. So we've been able to do this using this resource, and the reason it's no IRB approval is because we have IRB approval to bring the person in, draw their blood, but we send it to people around the world or around the country in a de-identified state. So they don't need IRB approval because they're not doing human subject research. So this is the gap is really a living biobank. It doesn't have people with disease. It may, but it doesn't. It's not focused on getting people with disease, and it allows us to do hypothesis-driven research about particular genetic variants in a normal population who either carry or don't carry those variants. And that's been extremely productive over the last 3, 4, 5 years. So these are just examples of variants that are all regulatory. There's nothing wrong with the amino acid sequence here, but it changes the amount of the gene such that it results in changes in B-cell signaling, in this case dendritic cell signaling, and I'm not going to go through these, another B-cell signaling with a molecule called BLK, which again gives us insight as to what these genes are doing are not causing disease. They're common in the population. They're causing an immune trait that in combination with other traits and environment predisposes to disease. So that allows us then to build models. This is just the B-cell differentiation pathway. Starts out in the bone marrow. We've shown that some of these actually influence the degree to which B cells that are autoreactive get out of the bone marrow. Others of them regulate the degree to which these B cells can get help from T cells, which is an important part of their differentiation in order to produce antibodies. So it allows you to build models about where this pathway really is dysregulated in the setting of autoimmunity, even though none of them on their own are abnormal. So we now are trying to expand this to a much larger population of a living biobank. And the reason for this is because increasingly it should be, I think, clear that actually it's the interaction between these different genes causing a particular, immune trait in a particular pathway that you want to get at. So people are now starting to ask us, gee, I'd like people with these 5 risk variants and people that don't have those 5 risk variants, and I predict that their T cells are going to go crazy, even though they represent, only a couple of percent or less of the population. So we're expanding it, and one of the wonderful things about being at the Feinstein Institute is it's integrated into this health system that's very large and actually has a lot of forward-thinking people in it because people hear this and they think, let's do it. So what we've done is figure out how to recruit people more efficiently. In the old days, we would register people, we'd have them fill out a consent form, we'd go to their house and draw blood. It was a very tedious and expensive process. We now are able to recruit from our employee base of 60,000 people. We've just initiated this last week, actually, so we're very, very excited about this. We have local community we could draw from, patients we can draw from. We've set online consent in process, and I'll go over that in a bit. And then we have a lot of these other things in the hospital, in the system. So just to go for the employees actually, they get a benefit, a paycheck benefit for doing the right thing for their health. I was asking before, why don't insurance companies give you a benefit for doing the right thing for your health? Well, we do that. We give you a few bucks off your paycheck if you go and get your mammogram or your checkup. So we have now agreed that actually participating in research is an important value for people in this health system, both as patients and as employees. So we give you that little benny if you'll enroll in the GAP. We also have this amazing call center that's in the center of Long Island, and I guess in the early days you were thinking about putting this call center offshore, and it is, you walk into it, I've been there 3 or 4 times now to talk to these people. You think of it as a call center as a really boring place where people are doing boring things. These people are really excited about this. They want to do new things. They want to help recruit for research, so we have now initiated several studies. The GAP is one of them, one on endometriosis, where basically when people are on hold for their 1 or 2 minutes, they get a message, they could be sent to a website and so forth. So we're just starting that. There were 500,000 calls last year? And it's going up. Tom was telling me we're now expanding this to the Department of Medicine, so it's really going to expand. So huge population resource, and those of you that do recruitment know that being able to recruit like this online with an online consent is just gold. So we also have a huge local community that we've also reached out to. So again, we have online consent that people get sent to. We do a little questionnaire. You're eligible to participate, and you're eligible to participate basically if you're over the age of 18. And you sign this consent, you download it. We don't have to send it to you by mail. It's automatically downloaded for you. You can, sign it online. And we also do this thing where we ask permission to do follow-up with a text message. Now, this seems like maybe it might be a trivial thing. But in fact, the important thing here is to get a huge population of people who are willing, and most people are willing, to participate as a registered person in such a resource. But we sent out about, 8 years ago, we sent out 30,000 letters to the then employees of this health system. How many did we get back? 300. And I know it's not because people aren't interested. People are very interested. I've gone out and talked to a lot of venues. People want to sign up. But when you send them a letter, it sits on their front hall chest. They open it up. Oh, that's a great idea. And then it never gets acted on. And the same occurs when we call back people who have particular genotypes that are of great interest to study, they say, yeah, they get the letter, they get the email, but, things move on. So we think that actually texting people, where people are more likely to answer a text, and they can do it immediately, and they can go to a link on the web to explain why you're asking them this, is going to be a very important part of making sure the callback rate, which is currently about 20%, will go up for this. So they get the consent, they say you're enrolled, they can go to any of 28 centers in the system, there's an automatic order. They can go and do it specially, or they can go and then do their routine blood draw a month from now. We'll get it. Comes directly to my lab. We can track all of this online. And so this part, the actual enrollment, is the big barrier, which I think we have solved. We'll see what happens over the coming months. Getting the blood draws, we have a central biorepository that's been running over a decade. It has millions of samples now. We do, obviously, high-throughput genotyping in my lab, and we have a very well-organized IRB and committee review for this. So, in fact, if there's a researcher in this audience who needs people of a certain genotype, you can email us. We have our online thing, and in 2 weeks we can get you approved and get you sampled. So, if we are able to actually get half of the employees of the health system involved in this, what can we do? So the first thing we can do is we can be much more responsive to the increasing number of investigators around the country and around the world who need access to this kind of resource. And we can also explore the genetics of other common phenotypes that we can either measure in the blood of those people, or we can do something that we can measure online. So the perfect example of, and I gather Niall is going to talk about this a bit, is PCSK9, which, as you know, many of you may know, it regulates LDL receptor function. There are some rare variants. Were originally discovered in the populations in the American Southwest by Helen Hobbs and her colleagues at the University of Texas. They found that people who had these variants, they were 2 or 3% in the population, had very low cholesterol, much lower cholesterol than the normals. And so they said, well, maybe if we interrupt this gene, it could be a way to treat cholesterol. It's 10 years later. These two drugs were just approved by the FDA for treating cholesterol, which is an unbelievably short time frame. But it shows you how going from rare genetics, like the TIC2 I showed you, to an actual treatment in a very short period of time, because you have a rare variant that in and of itself does not really help you with the population analysis, but understanding its function has a huge impact on what you might develop as a drug. I'm very interested in inflammatory markers in the setting of aging, and BMI was talked about today. I think those things are really interesting. I've just seen some data from a company where they're people with a high BMI that have lots of inflammatory markers. No surprise, they make a lot of IL-1, but there's a subset of that group that are low. Why are those people protected? Is that, a lot of the risk from BMI is due to those inflammatory factors. Maybe there's a bunch of people out there that are obese, but they're not really at risk. Maybe there's a genetic reason for that. Maybe there's an epigenetic reason for that. I mean, those are the kinds of things that you can ask about this. Another favorite one of mine is cognitive traits. So I've been working for almost 2 decades on something called absolute pitch. We're actually very involved in figuring out the genetics of pitch perception. We have iPhone apps for doing this. We've started having people in the gap run these apps. We have all the genetics. So it's really, you start to have this population where the genetics is completely defined, and we'd love to have whole genomes, and then you just do your phenotyping. It's much less expensive. It's a very different way of doing genetics, where you start with the genetically typed people and you ask very specific questions about phenotype. You either do that in vitro with a T cell, or you do it online with an iPhone app. And I'm sure you can think of a lot of other things to do, and I'd love to hear about them. So obviously none of this occurs without a huge team effort. This is my team a few years ago, but this does not capture all the people in this health system that are so responsive when I come and explain this to the call center or to the pathology department or even to people in primary care, they get it. We need to do this. We need to integrate information like this into the overall understanding not only of disease but of the human condition and human subphenotypes as well. So thank you very much.